restriction fragment length polymorphism. Restriction fragment length polymorphism is a difference in homologous DNA sequence that can be detected in the form of fragments of short length and large lengths. So these are the fragments of different lengths which are usually detected by the digestion of the DNA with the help of certain restriction enzymes. This technique is called as restriction fragment length polymorphism. Restriction fragment length polymorphism markers are heterozygous. Most of these markers, they are heterozygous and they are allele specific. In case of RFLP, probe is a labeled DNA sequence. This labeled DNA sequence actually attaches to different fragmented regions of the DNA. This is a complementary strand that attaches to those regions of the DNA where it is complementary. So with the help of a small probe that is usually labeled, we attach the probe with the fragmented DNA. That fragmented DNA that has been converted onto the membrane, which is called as nylon membrane. If we see that what is the methodology of RFLPs, that how RFLP is performed, the methodology is usually the genomic DNA is digested with the help of restriction enzymes. Once the genomic DNA is digested with the help of restriction enzymes, those fragmented DNA are separated with the help of a technique which is called as electrophoresis. With the help of electrophoresis, fragmented DNA, they are separated from each other. Once they are separated from each other, then those DNA fragments are transferred to the nylon membrane. Once they are transferred to the nylon membrane, then probe, which is usually a labeled DNA sequence, probe is exposed to the membranes where the DNA fragments have already been transferred. Once probe binds to the complementary DNA which is present on the nylon membrane, then this film is exposed to X-rays to study that what are the results of the restriction fragment length polymorphism. So this is the methodology how this technique is performed. To understand the phenomena of RFLP, we take here example of the RFLP. Here we can see that this is the DNA of an normal individual. Within this DNA sequence, there are restriction sites of an enzyme. The name of an enzyme is MST1. So there are three sites, site 1, site 2 and site 3. So if we digest the normal DNA, then with the help of this restriction enzyme, two fragments will be produced. This will be fragment 1 and this will be fragment 2. While due to a mutation that we can see here, this is the DNA of a person. In the DNA there is a mutation at a particular point. This is the point. So due to that mutation, the one of the site which was in the middle has been destroyed. So now there is only two sites for the restriction enzyme MST1, here and here. This has been destroyed. So if we digest the DNA of the individual who has disease, so the DNA of that individual will produce a single fragment. So here in case of normal, it was producing two fragments, but in case of the individual who has a disease, the DNA will produce a single band. That we can see here in case of normal and in case of disease. Here we can see that when we have run the DNA on the gel, we have seen that there are two fragments, fragment 1 and fragment 2. That is also here, fragment 1 and fragment 2 in case of the normal individual. While in case of that individual who has developed a disease, due to the mutation in that individual, this site which was present in the middle has been destroyed, so there will be only a single band in case of that individual who has the disease. So we can say that RFLP is a technique by which we can detect or we can study certain diseases, or we can identify the mutations in certain diseases. Here is another example of the RFLP. We can see here that how the DNA or the alleles, they transfer from the parents to the offsprings. So this is the DNA of the mother. This is the bands or banding pattern of the DNA of the father. This is the banding pattern of the DNA of the child. So these are the parents and this DNA belongs to the child. Here we can see in case of locus 1. In case of locus 1, this individual is a heterozygous. So father is heterozygous. Same is in case of mother. We can see that mother is also heterozygous. We can see 2 alleles. And if we do the comparison with the child, so child have got this allele from the mother, while the child has got this allele from the father. When we do the comparison at locus 2, this is the DNA fragments or banding pattern of the locus 2. In case of father, father is homozygous because father has just a single band. If there is a single band, it is called as homozygous. If there are 2 bands, then it is called as heterozygous. While in case of mother at locus 2, she is heterozygous. So if we see that what has got a child from the parents, so child has got this band or this allele from the father, while this allele from the mother. So with the help of restriction fragment length polymorphism, we can perform the paternity analysis. Here we will try to identify that there is a mother, she is a mother, this is the child, and there are 2 individuals, AF1 and AF2. AF1 for father 1 and AF2 for father 2. We have to identify based on the alleles of the child that who is the father of this child. If she is a mother, if we do the comparison that at locus 1, in case of child, this is locus 1. In this case, the child has got this allele from the mother. This is this allele. So if we check that this allele, it should be belong to the father. So which is the father from whom this child has obtained or got this allele? If we see in case of father 1, here the alleles are in relevant to this DNA fragment and in relevant to here. But if we see that in case of child, it is present at ladder number 1, 2, 3 and round about 4. So this allele has been inherited from AF2 because AF2 has this allele. So this is the allele of AF2 mean father 2. Now if we do the comparison for locus number 2, in case of locus number 2, the child has 2 alleles. One is this allele. So this allele has been inherited from the mother, while we have to see that this allele belongs to father 1 or father 2. If we do the comparison that this is actually the allele which is present in father 2 or AF2. So we can say that AF2 is the person who is the biological father of this child based on the alleles combination. So with the help of restriction fragment length polymorphism, that is a technique, this technique is used to resolve the paternity cases. In most of the paternity cases, restriction fragment length polymorphism technique can be used that can resolve the paternity cases. So restriction fragment length polymorphism is useful not only for the civil justice, it is also helps in criminal justice. RFLP used for constructing the genetic maps. With the help of restriction fragment length polymorphism, we can construct the maps of different genomes. So this technique is also used for constructing the genomes of different organisms. Disease resistance gene. In case of many plants or crops, disease resistance genes can be studied with the help of this technique. RFLP is useful for disease status. Many of the diseases can be identified based on this technique RFLP, like the most common diseases in the humans, Huntington disease, cystic fibrosis, and sickle cell anemia have been studied for decades with the help of restriction fragment length polymorphism. Restriction fragment length polymorphism also helps for the detection of the mutations which are present in certain individuals and causing the diseases. So we can say that restriction fragment length polymorphism have many uses, and mostly this technique is used for identification of the mutations in the individuals that have certain hereditary diseases.